To the last part of today's lecture, part 4 out of 4, the categories of mammalian cell culture. This is very important to understand. There are two major types of mammalian cell cultures. One is known as the primary cell cultures, and the other one is known as continuous cell lines. So for primary cell cultures and continuous cell lines, they have a lot of difference in between them. Primary cell culture is the dissociation of cells from a parental tissue, as well as we can also include plant tissue through enzymatic or mechanical measures. Uh, when we talk about plants, we excise them through mechanical means. But in case of mammalian cell culture, we normally use an enzyme which is known as trypsin. So trypsin, what does trypsin does? It loses the cells from the other parts. It cuts the connections. And then we maintain the growth of these cells in suitable substrate with a suitable substrate of either glass or plastic or other common novel polymer materials under controlled lab conditions. So these cells are directly taken from the fresh tissues. Family cells mostly represent the tissues of origin, and they are taken directly and processed to establish under lab conditions. They are derived directly from the tissue, as I said, and they're not modified. This is why they are very much similar to the in vivo state of the cells. This is why they exhibit the physiological or the normal physiology of the cells from where they're taken. And since they exhibit the normal physiology of the cells like in vivo state, they are they're providing to be excellent model systems for the studying of normal physiology, biochemistry of cells, for example, metabolic studies, aging, signaling studies, effect of drugs and toxic compounds on the cells, etc., etc. But the problem, the major disadvantage with these cells is the fact that they have a limited lifespan, which means they will divide only a few number of times in the cell culture. And eventually, after certain number of division, they will reach senescence. Senescence means they will stop dividing and they will die. This is why they are more difficult to culture and maintain than a continuous culture. Here I have mentioned some cell lines. So the most popular primary cell lines are epithelial cells, fibroblasts, keratinocytes, melanocytes, endothelial cells, muscle cells, hematopoietic and mesenchymal stem cells. Now, these are actually primary cells. The primary cell lines, we have said that they will be dividing a few times, a certain number of times, and then they will lose the capacity of division, and senescence will take place. But we can actually, these primary cells, we can maintain them. These primary cells can be manipulated for indefinite times by subculturing through in vitro processes, which is known as transformation. Means in transformation, we can change the primary cells to become continuous cells. Or another name for primary cells is mortal cell lines. They're mortal. Mortal means after a number of divisions, they will die. And the continuous cells, we can call them immortalized cells or immortal cells. So we can transform a mortal cell line to become immortal through some specific chemical stimulus, or even there are viruses. So if we culture them with some viruses, infect them with certain viruses, they will become immortalized and they will become continuous cell lines. Now, when a primary cell line, it transforms to become to continuous cell line, they can divide indefinitely and they can become immortally, they can become immortal, continuous, and secondary cell line, secondary or continuous cell line. Once they transform, they are no more primary, and as that change is irreversible, and they will be immortalized. The next type is continuous cell line. These are the cells that acquire the capacity for infinite growth. Now, as I said, they are the continuous cell lines can be by already by nature. They are continuous cell lines, immortalized cell lines, or we can take primary cell lines and we can transform them and we can become them immortalized or continuous cell lines. Immortalized cell lines are either tumor cells. Tumor cells means they are cancer cells, and they do not stop dividing. Or these cells, either they are cancer cells. So by nature, mammalian cells should be primary cell lines if they are normal cell lines, and they will be continuous or immortalized cell lines only if they are cancerous. Or these continuous cell lines, they have been artificially manipulated to proliferate indefinitely, and they can be cultured over several generations. So because these immortalized cells, they can grow and divide non-stop, continuously, they will eventually fill up the dish or the cultured flask or any other culture whether we have, then in which we grow them. Another thing about these immortal cell lines or cancer cell lines, you can actually, they are anchorage independent cells. So if you have a normal cell X in a mammalian organism, it will be anchorage dependent because it will, until unless it's primary cell, it will be anchorage dependent. But when it becomes neoplastic means when it becomes cancerous, when it becomes tumors, then you can culture them in suspended form. They are no more anchorage dependent. You can see the drastic changes come to a normal cell when it becomes cancer. It loses so many capacities, it gains so many attributes, they become so dangerous. And of course, they lost their sensitivity to factors associated with growth control. And they also lose their anchorage dependence, as I said, and they're very easy to culture. Now, when we are working in cancer studies and we are culturing cancer cell lines, now the cancer cell lines, they are continuous cell lines, and they are immortal cell lines, and they are anchorage independent. It means you can culture them under suspended conditions. In a way, this is a good thing because in our investigation studies on cancer, we do not need any solid supports. We do not need any scaffolds. So you can culture them just like plant cells or just like microbial cells under suspended conditions for all of the investigations. Either it's genetic or it's therapeutic or it's metabolic or it's metabolomic, genomic, any kind of studies, these cancer cell lines. And they're immortal. So they won't ever have senescence. They will divide, and no problem at all. The advantage of using continuous cell lines, as I discussed, they can be cloned. You can clone them, and then you can easily isolate the mutant cells. You can do investigation of growth. You can use them for prediction of hybrid cells in biotechnology, and for the production of pharmaceuticals, vaccines, hormones, other chemical therapeutics, growth factors. Now, what we had been discussing until now was all about culturing the cells in 2D. If you have a flask, or if you have a vial, or if you have a petri dish, this is 2D. In the recent times, the mammalian cell culture have been added another dimension. And what I'm saying is not figurative, it is literal. So from 2D, another dimension means 3D. Now we are culturing cells in 3D. Now you could see this is a mammalian cell culture cultured in a 2D culture vessel. And this is the same cell cultured in a 3D scaffold, and you can see the difference. The 2D, the cells when they are cultured in a 2D manner, 2D culture system, look like this. a monolayer. But when you culture them in 3D, so you could see they are in a 3D spheroid shape. In 2D culture, plastic is used as a substrate, as a polymer. They are unable to mimic in vivo micronutrient and like cell-to-cell communication. This is a very important point because when you culture cells in 2D, it is said that 50% of the cells will stick to the substrate, which is a Petri plate or a flask or any other culture wire. 50% of them will stick to the growth medium, which means the cell-to-cell contact is going to be really, really reduced. And when the cell-to-cell contact is reduced, the cell-to-cell communication is interfered. And when the cell-to-cell communication is interfered, you will never mimic an in vivo microenvironment. And you don't know if what are you deducing from these experiments is reliable, because you are not giving the condition in which they are in a natural environment. This is why a 3D culture, it mimics the micro environment that is in vivo, and this natural extracellular matrix is not plastic, it's some other materials, and it's in 3D. This is in 2D. 2D cultures are, of course, the cells lose their morphology because when you culture them on a flat surface like this, they tend to grow along the surface, the flat surface, and this really interferes with their native morphology. Like, look at this cell, the morphology of this cell and this cell is one cell cultured in two different ways. This resembles the in vivo, mimics the in vivo way it is growing in a living medium, living organism, and this is in a culture. So you could see when these cells, they look stressed. They're not happy as this cell. That's why the new technology is the 3D. It's a little more expensive. You have low throughput capacity, but it's indispensable for tissue engineering, regenerative medicine. So the main problems in the 2D, as I discussed, the cells that cannot grow in third dimension because of the surface, the flat surface, as well as the gravity. The gravity pulls them. And when this happens, there's limited cell communication. And when there's limited cell communication, it does not really mimics the in vivo scenario. And not relevant to human physiology, as we said, because in the human physiology, the cells are cultured inside our body. They are in 3D. In this condition, you do not know what the cells are doing is exactly what they would do in a normal in vivo environment. But in this condition, it's more likely, what we cannot say it for sure, that in this condition, they will behave exactly the way inside a living system. But this is more likely to be mimicking the in vivo environmental conditions, or the in vivo conditions, not environmental. So they highly resemble an original tissue, and it can be very easily extrapolated with that of in vivo response. We can use the arrangements for a wide number of cellular functions like proliferation, differentiation, signaling, gene expression, etc. As I said, 3D culture, the new way to culture mammalian cells, and they are more likely to represent a perfect substitute for the culture of cells to have better insights, more resembling towards the in vivo types of their natural environment. Okay, this is something I got from the internet. This is polystyrene. It's a porous polystyrene scaffold, and this is a 3D scaffold made by a company recently. So you could see this is in 3D. Says 200 microns is the diameter. And when you culture the cells will eventually permeate freely into the scaffold. So you can see the cells were sprayed here or cultured here with a growth medium, and eventually they start coming down into settling into these three-dimensional porous structure, and the cells now can form a complex tissue-like 3D structures. So you can see all X, Y, and you cannot see, but of course, the screen in which I'm recording is already in 2D, but the representation is in such a way that you could see that the image is 3D, and the cells topology as well as the morphology looks really better than these flat monolayers. And we are of the opinion, the scientific community is of the opinion that culturing in these 3D scaffolds is more likely to give a better mimicry of these cells, how they grow, how they differentiate and express themselves in a living environment. So that's why these days these 3D structures are preferred. It's the new technology, and within the next few years, I think the 2D cell culture would become uh, antique. It will just disappear. But again, one thing, the closing remark of today's lecture, that this 3G cells, the 3D cells culture scaffold is only going to be for the anchorage dependent. If we have an anchorage independent cells, we don't care. It won't change much. This was all for today's lecture, and I will just stop it and share it with you guys. We will have our questions in the interactive session.